Neuroblastoma and Histone demethylation. Definition. A neuroblastoma is a form childhood cancer that forms in nerve tissue and usually begins more frequently in the adrenal glands that are located in the upper part of the kidney.
Learning medicine is hard work. Osmosis makes it easy. It takes your lectures and notes to create a personalized study plan with exclusive videos, practice questions, and flashcards, and so much more. Try it free today. Neuroblastoma is a type of tumor composed of neuroblasts, specifically neural crest cells, which are cells involved in the development of the sympathetic nervous system. Neuroblastoma is the most common cancer in infants, and it's only rarely seen in children over 5 years old. When a fetus is in its 5th week of development, special cells called neural crest cells start migrating along the spine. In the thoracic region of the spine, neural crest cells differentiate into the neurons of the sympathetic chain, lying on either side of the entire spinal cord. In the lumbar region, neural crest cells differentiate into the cells of the adrenal medulla, the inner part of the adrenal gland that sits atop the kidneys. Together, the sympathetic chain and adrenal medulla form the sympathetic nervous system, connecting the brain and central nervous system to various organs including the heart and blood vessels. So,
Application in nursing. The disorders that are performed by the nursing staff in the perform anti-HD2 immunotherapy. This treatment increases survival in the patients with high-risk neuroblastoma by 20%.
Neuroblastoma is a word no child should know. This is the tumor that steals the child. It absolutely steals the child. Neuroblastoma is a cancer of the autonomic nervous system, in particular the sympathetic nervous system. It's historically been very difficult to treat high-stage neuroblastoma. So generally it starts in one spot, let's say the adrenal gland, the gland just above the kidney. With the development of this tumor, it has the capacity to spread. The body should see this tumor and try and destroy it, but the tumors are too smart for the immune system. It requires new biology, a new understanding of the disease, better surgical techniques, immunotherapy, targeted therapy. So all of these things are what's going to move the needle in neuroblastoma. The St. Baldrick's Foundation has been
Pros and cons. There is no type of benefit since this disease involves very serious consequences for the child who is detected with this pathology, since its mortality rate, since they are aggravating tumors with a high mortality and great involvement of the general condition.
Welcome again guys, welcome to another video from Shomo's Biology, and in this video we'll be talking about the histone acetylation and methylation. I have different videos on entirely dedicated to histone acetylation, deacetylation, and also about the DNA methylation process in epigenetics videos. But in this video I'll be talking briefly about both of these processes in a little bit details, because I've seen some of the students have problems regarding the acetylation and methylation and what they actually do. So for that clearing up, I'm doing this video. So let's say histone acetylation, and why we require all these things. This acetylation, methylation, these are chemical modifications, obviously, because we have histone and we all know what is histone. If you don't know what's histone, definitely you should not watch this video right now. You should watch my histone video previously. I hopefully put a link here in the description and also in the annotation. Click here and watch that video first and then come back here and watch this video. So I'm assuming that you all know what histone is. Let's say for your convenience I'm drawing it again. So let's say here Histones are protein molecules, one of the very common protein molecules that are present. Let's say these are the histones. Let me draw it this way. These are the histone, for example, and we have also the DNA in the red color here. So DNA is wrapped around these histone molecules out there, wrapped around histones. Why? Because we have a very compact structure which is called as a chromosomes. Remember, we all know very much complicated structure. If we look at zooming into the chromosome, we have the 30 nanometer fiber, then we have all those beads and string model. This is called the beads and string model because this is a string as a DNA, and the beads here are acting as a histone where the DNA is wrapped. So these are histones, and that is the DNA. So this is how it presents. Now these histones are protein molecules, and actually there are 4 different types of histones are present. They are arranged with each other and they have different terminal, N-terminal as well as C.
Issue apply in Ecuador. In Ecuador, childhood cancer has been more common in 13% in recent years because the symptoms are so vague that 50 to 60% of all neuroblastoma have spread metastasized to other parts of the body by the time the diagnosis is made.
Okay, so now we're going to go into more specific detail about histone methylation and histone acetylation and the consequences it has on expression and how these different histone marks are actually read by the cell. So histone acetylation is universally associated with gene activity, so an active gene that's being transcribed. This is easy to explain in an electrostatic way because when we add an acetyl group to a lysine of the histone tail, this neutralizes part of the positive charge of the histones. And so the histones are not as strongly attracted to the negatively charged DNA, and therefore it's going to be more open and more accessible to the transcriptional machinery. But the second mechanism by which acetylation can be associated with transcriptional activity is more of a general mechanism that's true for other modifications as well. And that is that this acetyl group can be bound by a bromodomain. So a bromodomain is found in many different types of proteins. And it's this bromodomain which themselves recognize the acetylation group, and one of 2 things can happen. Either the bromodomain itself has other components within that particular protein which bring about changes. Say, for example, they're chromatin remodelers, which we'll deal with in another lecture, so they actually move the nucleosomes around. Or the bromodomain might recruit another protein that can do that. This is a little bit similar to
Conclusion. Neuroblastoma are the most frequent cancerous tumors during the last decade that occur frequently during the first 30 years of life, which are mostly detected when performing a check for some type of stroke.
In the previous video, we discussed about the CpG ionides. If you want to watch that video first, you can click the link from I button or find it in the description. Now, in this video, we'll be discussing about DNA methylation in detail. Let's get straight to it, what DNA methylation is. It's the transfer of methyl group from same molecule to adenine or cytosine base of DNA. So we see here it involves the addition of methyl group to the DNA bases, and this addition takes place on 2 DNA bases, that's adenine and cytosine. So we can say methylation takes place on 2 DNA bases among 4. Now what mediates the transfer of methyl group? It's done by an enzyme, namely DNA methyl transferases. In short, DNMTs. And these DNMTs are of 3 types: DNMT1, DNMT3A, DNMT3B. First of all, let's see what's the specific function of DNMT1 enzyme. This DNMT1 has capability to maintain the pattern of methylation within the cell. So this is for the maintenance of DNA methylation. But what does maintenance mean here? Suppose we have a DNA molecule, it has 20 bases methylated, of which 15 are cytosine methylated and 5 are adenine methylated. And it's during the replication of DNA molecule, the newly synthesized DNA molecule is methylated at same 20 positions, with